Aside from his celebration his celebration that we give for him as a founder of computer science and his work at Bletchley Park on code breaking, Alan Turing wrote a remarkable seminal paper in mathematical biology. It was written in the last 2 years of his life, in fact, and it was really a surprising, remarkable paper. It was on morphogenesis, on the development of plants and animals for at least certain parts of development of plants and animals, and it led to his coming up with a set of partial differential equations, nonlinear partial differential equations, which he applied in certain special cases to pattern formation in leaves and gastrulation of the ovum. It's essentially a model which is of you might say, of a hydrodynamic type, where you have waves of morphogens, which are chemicals that diffuse between cells, in his theory, and they form constructive and destructive interference patterns, so you get a resonance. So for instance, on an annulus, he found you get a symmetry breaking in which you get, starting from a uniform set of morphogens, you end up with a pattern of alternating concentrations and so on, which might be the basis for sprouts coming up in particular plants. This would be an annulus, which is a cross-section of a plant, such as he mentioned, a hydra, and so on. And he gave various such examples. This was largely ignored initially, but it's become a very prominent part of mathematical biology. And one of the most authoritative people in this field is James Murray. There's a book by him called Mathematical Biology in 2 volumes, which has very beautiful examples of color patterns in animals. And it becomes quite clear that just like, for instance, in a drum, where you get, when you have Chladni figures, that the boundary becomes a very important part in solving this wave, they're essentially a kind of wave equation. In any case, he's done written many papers on this and other subjects in mathematical biology. So James Murray was a professor for many years at Oxford University, where he started a center for mathematical biology and, as a founder and also director. And then, after many years, he went to Washington State University in Oregon, and he's now an emeritus professor here. And his talk will be on mathematical biology. Let me read it. It's probably up here. Mathematical biology, past, present, and future. From animal code patterns to brain tumors to saving marriages. Yes. Professor Murray.
Can you hear me okay? No. It's switched on, so it's okay? Yeah. Okay. Well, it's certainly a pleasure and a great honor to be invited to give a talk at the Turing Centennial meeting. Certainly, Turing wrote really a seminal paper on pattern formation generally. When one reads his paper, one realizes that in the beginning of the 20th century, really there was a lot of activity going on in the applications of mathematical models in biology, medicine, ecology, and so on. But one paper that practically nobody mentions is by Daniel Bernoulli, the mathematician, who wrote it in 1775, and it was all about the effect of vaccination. It's an important paper, and when I complained about this in Oxford once to a group of people, and they said, well, it served them right, he wrote it in French. And so, anyway, let's see if I can. Okay, so what I want to talk about are just three things that I've been involved in, and the first one is really associated with Turing's paper, but as I said, there was a lot of activity going on before Turing's paper was rediscovered, and there was some Russian people who actually produced experiments that produced a spatial pattern, a man called Belousov, and he couldn't get it published because they thought this is a ridiculous field. But then that's what my first postdoc thought about me moving into it. Anyway, my whole approach to this is not very mathematical, and I really go along with really what Benjamin Rush said, that it really is of little use when it's only confined to speculation. And when you collaborate with biomedical scientists, they actually want to know something that they're doing, that they're interested in. And it's why interdisciplinary collaboration, genuine stuff, is really very exciting. Actually, Rush, he did his doctor's degree in Edinburgh at the time of the Edinburgh Enlightenment. It must have been very exciting. And so he was exposed to, the sort of this intellectual excitement, and no doubt the prejudices of a typical Scottish Calvinism. But that's just part of growing up in Scotland. Okay, so let me talk just very briefly about the thing that got me interested really in Turing's seminal idea that basically what it says is that if you take certain chemicals, two of them is enough, and they react, one is called an activator, one is an inhibitor, and they diffuse within a finite domain, then if they diffuse at different rates, you can generate steady-state spatial patterns. And at the time, I thought this was a very exciting thought. Nobody had come up with it. Well, Belousov did. I didn't know about his experiments at that time. And so someone asked me, how did you get involved in animal coat patterns? And they said, well, of course, it must have been Turing. Actually, it was our 5-year-old daughter. I was reading her Kipling's Just So stories about how the leopard gets its spots. And at the end of it, she said, but how do they get their spots? And so I said, oh, must be known. I'll ask colleagues in the university. And no one did. And I still don't. Anyway, this is just a nice picture. And that was one of the fun things about working on animals, that you came across all sorts of lovely photographs. And so what I decided to do was to really take Turing's ideas of pattern formation and see if somehow or other it was associated with animal coat patterns. And when I got involved in this, this is a pattern. This is really the two partial differential equations that I used. Turing's ones were more complicated, but in essence, that's what they are. And this, you don't need to know the details. That's really not at all crucial. This gamma is just a scale thing, a scale parameter. And the reason I took this one was that I had been in France, and there was Daniel Thomas in the University of Compiègne in 1975. actually came up with specific chemical reactions that produce spatial patterns. And what I found really surprising when I met him in Compiègne, and I said it was, you know, this was the first experimental system that actually confirmed Turing's theory. And he said, Turing? He had never heard of Turing, other than during the war. And so this was an experimentalist who came up with patterns in a reaction. He hadn't heard about Belousov either. Okay, that's just a nice picture that's got patterns of a cheetah. Okay, so let me talk about what the model produced. So I started thinking, supposing I take a domain like this, and so I solve the equations, and they produce spatial patterns which are shown in these black things like this. And so I didn't vary any of the parameters except scale and geometry. And so it wasn't playing around with lots of parameters. And so what one found just, even at this stage, was this: if the domain is very thin, just as it is here at one end and thicker at the other, you can have patterns going around the tail, and then you end up with stripes. And so really one kind of came up with the thought, well, you know, you can have a spotted animal with a striped tail, but never the other way around. And this actually is an animal that's not very well known. And the reason I show this one is this Geoffrey Saint-Hilaire was really an incredibly original biologist who wrote a book about teratology in the early 19th century. And he traveled in South America, and he came across this cat that is now called it. This is just a typical spotted animal with a striped tail that my friend Andy Dobson gave. And then, of course, if you look at other ones, it really is kind of universal, almost. And there are lots of friends who have been trying to find animals that are striped with spotted tails, but they haven't done it yet. Okay, so let's just go a little bit further to be more specific biologically. If you look at zebras, then this is what a zebra looks like at just a few days apart. So if there is a mechanism that is activated when the embryo is like this, and it produces a spatial pattern of chemicals, which we can call them morphogens, because then they affect the cells in the skin, which produces melanin, and then produces the spike, the marks. And so if they started it here, then you end up with this kind of zebra. And this is typical pattern between a leg and a body. They are totally unique. And this is one of the things that comes out of the model, is that any initial condition where the patterning is going to be started, they're going to be minor and mild, small, large, but not large, but small variations. So every animal pattern is unique. And of course, this is how they recognize each other. I did a little bit of computation to see what would happen if I took a domain like that. And it's not very interesting, but what it does show is that you do get this sort of patterning. This sort of thing one can see where the tail, the back of it rather, is swiveled round. On the other hand, if the embryo is just a little bit bigger, and it's the same mechanism, then you can get more stripes in. And so in that case, what you get is one of the other kind of zebras. And of course, this was all done in the kind of early 70s. And then came later on, there was the genetic revolution in which genes are the answer to absolutely everything, except for the stripes on an alligator. And how it came to dinner conversation in my college in Oxford, and we were sitting drinking the port, and one of this visitor worked on alligators. And I said, oh, well, they've got stripes. I've been doing some things in stripes. And he said, it's interesting, the male has one more stripe than the female. And there was another visitor. They said, it's genes. It's clearly genes. And so I got very involved. It turns out temperature determines sex with alligators. And the sex of males is a little higher. And so we came up with the idea that you incubate an alligator so that it's a female and change the temperature and it'll grow a little bit bigger. And we predicted it would have one more stripe, which is exactly what it did. I couldn't remember the geneticist's name, unfortunately. Anyway, so just very briefly, I then looked at things like this, where the scale factor between that and that is about 1,000. If it's very small, you can't get any pattern. If you then have the first bifurcation, you get there's quite a few of these. The next one, of course, these are belted Galloway cows, which were quite common where I grew up. And now that's just a nice anteater that I happen to have. Okay, just really the last thing is this whole business of size. This is a very small embryo. This is enormous. And the reason for that is that the boundaries are so far away, they can't really affect the pattern from a small point of view like that. So the argument is then very small animals are uniform in color, so are very large. And I thought I'd take a mammoth because it must have been uniform in color. But if you look at elephants, I'm doing well. Can you hear? Yeah, still okay? Okay. I've lost the button. Oh well, okay. And then you've got the gradation in between. And the gradation in between, you would have things like the giraffe. And there are two different kinds, of course, of giraffe. And sorry. And so what you get is you can generate the same sort of patterns between the two different kinds of giraffes. So the manipulation is, as I said before, it's not a lot of parameters. It is really just scale. And so that would be a very efficient way of really generating animal coat patterns if you just had one mechanism and you just vary the size. Of course, it is a touch simplistic, and that's really why this photograph that our daughter sent, it clearly disapproves. So can I just take this and put it back? I'm having to hold the battery in with my finger. It's okay, John. Yeah. Okay, what I want to talk about next is a bit on brain tumors. And that's one of the nice things about mathematics and biology. There is nothing sacrosanct. And you just work on whatever either colleagues interest you in, or you get excited about, or what comes out over dinner, or whatever. And this was a neuropathologist who approached me to see if I'd be interested in working on brain tumors. And the aims of this are really, one was whether we could come up with a model of growing brain tumors, specifically the tumors called glioblastomas, which are always fatal and always awful. These are some colleagues down at the bottom. It's Buster Alvor, and then the others are graduate students and postdocs of mine. I always used to make them get involved in things other than their thesis. And so the aim is we want to try and really make it very practical. And so I'll highlight these limitations of really imaging techniques. We want to determine really when brain tumors started. And in spite of all the what's going on about cell phones, there's certain evidence increasing that there are effects on the brain with cell phone proximity, and there are some experiments being done by colleagues in France. I also want to quantify patient treatment before it's given, because I mean, it's a bad enough disease as it is, and the treatments are one doesn't know, and so we'd like to quantify that. And then just a little bit of history to do it. People have been doing trepanations on the skull really for thousands of years. And this is an Inca skull where the person has had 4 trepanations where they've gone into the brain, and they've all healed. However, more recently in Kenya, the Kisi tribe do it by having them sit on a chair just outside, and they scrape it to relieve what they think is the problem pressure or whatever. And of course, these trepanations were very common in Europe, really up until about the beginning of the 19th century, when the people then went into the hospital to have them done, and of course most of them died of infection. But in the case of the Kisi tribe, the mortality rate is, to first approximation, 0, which is really very interesting. But then I thought before I describe the model, the history of medicine, of course, is very long. The major, without any question, the major figure in early medieval times was Henri de Mondeville. And he was a surgeon to 2 kings, etc. And he wrote a book in Latin on surgery around about that time. And he was very straightforward in what he thought about the medical world. And he said surgeons are, as you could read them, illiterates, bumpkins and barbers, fellow travelers, vagabonds. It really stretched my Latin, I can tell you. Body wenches, ruffians, and brothel keepers. And it really, he just was, I mean, he was a man of his time. I mean, people came, he could, simulate virginity, I mean, at a price, etc. And he did all sorts of things, but when it came to medicine, he really had ideas that were really hundreds of years before his time. And so it's not surprising he was a major figure. Okay, let me now come and actually talk about real brain tumors. This is a typical, the MRI, whether they're called T1 or T2, are CAT scans of the brain. And people with brain tumors end up usually going to the hospital because there is some unusual effect. They fell down, they lose their speech for a while, or anything can happen. But if it's peculiar, then they go to the hospital, and if they think it's possibly a brain tumor, they do a CAT scan, and there are 2 different kinds. This one is less accurate than this one, but basically, if this is the kind of tumor density, and this part in the middle are necrotic cells, then this one is much more accurate. And so, when one wanted to make a model, one thinks it's immensely complicated. So what do you do? And really, I suppose it's been my philosophy, you start with what you know. And what we know are there are cancer cells that multiply, and these cancer cells infiltrate the brain. So what we thought we'd do is we'd construct a model that said if you go to one part of the brain and look at the rate of change of the cell density, the cancer cell density, then there's going to be a contribution from diffusion, there's going to be a contribution from proliferation, and we just wrote down the mathematical equation for this. And so the mathematical equation is just the shorthand version of this. It encapsulates more. It says that this is the diffusion coefficient, and it varies whether it's in grey matter or in white matter. It's slower in grey matter. This term represents really the proliferation of the cells. And of course, the mathematicians in the audience will say, but you know, that means that the cancer grows exponentially. And sadly, it does. And I said, originally, we have to put some limit on it. And pathologists, neuropathologists are different. They see people in autopsies. And so they have to detach themselves. And he said, no, you don't have to worry about that because the patient dies long before any limitation comes in. And unfortunately, that is indeed what happens. When you take a CAT scan of a tumor, it is immensely complex. I mean, you get a millimeter range, but what you can do is you can work out what the volume of it is. And so what we thought we'd do was then work out the volume and then look at a sphere growing in the brain, a tumor sphere growing in the brain, with the same parameters. And you say, how we get these parameters is you have 2 CAT scans, and then using the model, we can evaluate the diffusion coefficient and how fast the things grow. And so, but the equation then is much simpler. I mean, this is sort of second year undergraduate mathematics can solve that. And the solution is down at the bottom here, where D, if you make that constant and that constant, then you get something that looks exponential, and R is the distance from the center of the tumor, and N are the number of cancer cells where the tumor started. And of course, one question is, you know, what's the value of N? And when I talk to the medical people or the scientists who work in it, and they say, well, one of them said, well, it could be 5. And I said, really? And his colleague said, no, it could be 5,000. They don't know because they don't know how many cancer cells you need before it starts to spread. Okay, so what? Of course, once one thinking back to Benjamin Rush, here we've got a model, we've made it very simple, we've got a solution. What one would really like, and what the medical people want to know, and much more importantly, what the patients would want to know, how quickly is it going to grow? And that's what from the solution, we can show that the diameter of the tumor is just the square root of the 2 parameters times the time from the time it was observed. I mean, a ludicrously simple formula. And then you can work out the velocity of growth. We just differentiate it, and that's the velocity down at the bottom. And of course, that's all very well, but we really must compare it with data. And this is data that we got, these results were obtained just before that, and Mondane and his colleagues were in Seattle Medical School, and these are low-grade tumors. The data from a large number of them, they are untreated. The problem with glioblastomas is that the surgeons always want to do something about it. and the family wants to do something about it, and we probably all know people who have had these terrible tumors. There is pressure from friends, family, everybody to do something. This was the only untreated tumor glioblastoma, that we could actually find. Okay, so then that's really the background, the simple model and the basis, and this is the first kind of comparison with data. But this is what the brain looks like. And this is a remarkable website put out from people in McGill. And what they have is they have millimeter slices of the brain, of real brains. And so what we did is we now take these real brains and simulate the model. And this is this is that one untreated brain tumor. And that is at diagnosis. And this is, well, you can see the dates, September, and this is July. And this represents what it was at autopsy. This was the model simulation, and this was the post-mortem MRI scan. These lines are lines of the same concentration of cancer cells. And this is, and the cancer started there. And this was this was September, which was just after the patient died. And so one can immediately see the problems with current imaging techniques. And the problem is that this black line is where you can actually see the brain tumor in the most sophisticated scanning technique or any other technique that is currently available. And the threshold of it, I mean, is really a large number of cells, but one can see that the tumor has grown way beyond anything that you can possibly see. And this is just a kind of movie that we made of really the growth of a cell. I mean, we took parameters that were really among the more severe tumors, where the median life expectancy is about 6 months. But until you see a black line, the tumors, you still can't see it. And so in about a few more days, and there you can start, and that's what you can see. So the problems really for the medical world are really major. For the patient, they're major. All one would like is that the medical world would really think about it and not just do what they always do. But then, I mean, you know the joke about the difference between a neurosurgeon and God is that God doesn't think he's a neurosurgeon. And so what I thought we'd look at the time was that it's very old data, but it's very interesting. 57 patients lived for 39 weeks after surgery, and 58 patients didn't have surgery. It's called resection, and they lived for 32 weeks. So, but the question is, what are these 7 weeks? What are these 7 weeks like? And anyway, and so what can the model suggest? I think it's already clear why surgery isn't going to work. And so let's look at one example of this is what the surgeon sees, and the surgeon tries to cut out a little bit more than he can see on the scan that he has available. On the other hand, if you take the scan of higher resolution, in other words, the kind of mathematical model that can actually move whatever level you want once you know where the tumor is, then this is what it looks like. And the parameters we used were really typical of typical patient parameters. This is the recurrence that happened with this patient about 6 months later. And one of the, in quotes, mysteries, according to neurosurgeons, if you think I'm knocking them, yes, I am. Because they do not normally listen to science. When I said to one of them, why on earth do you do surgery when you know it's not going to work? And he said, well, if I don't, somebody else will. And that, I mean, is in keeping with Henri de Mondeville, but he was also a good doctor. One of the mysteries was, when you do surgery, it recurs multifocally. In other words, it doesn't appear in just one place, it comes from all over the place. But of course, if you look at it with the most, a better scanning technique, of course it's going to recur all over the place, not just near where it is, but it could recur anywhere in the brain. And so, really, one wants to ask questions that patients want answered. And if you take a typical patient, 2 scans, and these are the values of the 2 parameters, the values don't matter, but diagnosis is typically when the tumor is 3 centimeters in diameter and the patient dies when it's about 6 centimeters. And so one can use that simple mathematical model and you can actually get an estimate of the survival time depending on these 2 parameters that you get from the patient. And so, but once again, we've got to compare it with data. And this is data, and some of it came out in 2010, but this is survival time in weeks and the percentage of people, of patients. And this old data by Kreth gives one curve, and Bob Rostamale in the medical school in University of Washington did the other one, and the other one is they calculated the predicted survival. And so this simple model does seem to really, it's got certain reasonable comparisons with real patients. And so what we would like to do, of course, if there is there will inevitably be major clinical studies on cancer as a result of cell phone use. And so the question is, how do you find out when the tumor started? Well, you just take the solution, and it's a sort of, and you invert it, and then you work out and say, well, if R is, you know, you just take this and say, depending, well, if you start here, at R equal to 0, the tumors grow to a detectable size of 15 centimeters. And then, if you're given R at detection, what you can do is you move the solution backwards. And what that does is it then produces an expression. You solve this equation for a given R, given parameters, and you work out the time. And this is the sort of thing that, you know, so one can do it in, you know, in MATLAB. Took me ages, which is why I had lots of students do computing for me. Anyway, so this simple model can give other estimates. And so basically, what you have is, if this is from that little film, if this is what is observed, then what you can then do is you can move the thing backwards and you find out not only when it started, but where it started. And so that then will tell you when and where the tumor started. But then there are other questions that came up. I mean, why do some people live longer than others with the same treatment? Well, turns out if you start off a tumor with the same amount of tumor cells and let it diffuse according to that model, then depending on the diffusion coefficient and how quickly it grows, it turns out that the diffusion, if the diffusion is very small, it's very, very steep. And not only that, the width of it depends. If D is very small, then it's very sharp. So when a surgeon tries to cut out a tumor that's got a small diffusion and a small diffusion, it's very much more contained. Doesn't mean they're going to get it all, because there's always a long tail. And in fact, that's really what it kind of looks like in much more detail. These numbers that I've taken are really all from patients. So let me just summarize. Basically, it negates these long-standing things. It can suggest new ways of gliomas. You know, we can tell when it started, etc. We can see why some treatments work better than others, and look at treatment efficacy. And finally, what we're currently doing is getting a lot of data from really several hundred patients. Let me just finish up the brain tumor stuff, going back to Henri de Mondeville, you know, who really felt very strongly about his subject. And he said that surgery is surer, more preferable, more noble, more perfect, and more lucrative. And that, which he ended up by adding. And he really wanted to do that. Okay, let me talk. I'll go through this fairly quickly. And again, these are a bunch of my former graduate students, and John Gottman is a psychologist who really got me involved. And so what we did is, we were interested in whether we could quantify a marital interaction discussion and then make predictions about the stability or longevity of their marriage. And so what we came up with was that the couple, and maybe I should add there were around 700 of them, who came to the lab, and they really talked about a problem of contention, and we filmed it, and we scored what the husband said and then the wife said and so on, and then we got data. And so really this the series of positive numbers minus negative for what they say. And so one had to have a system of scoring what the wife and the husband said. I gave a talk about this, it's now 3 years ago, and it got into the Sunday morning, and whoever wrote it then came up with this cartoon where, you know, you've got to choose a topic of contention to discuss. And so then they said, divorce. Anyway, so how you score it is a very simple system, where affection, depending on what it is, contempt is the most corrosive thing in marriage. And behind a one-way mirror, and the couple knew this was happening, is that there were a bunch of graduate students who scored what they said, and then they came up with what each of them said with a specific number. And then what you do is you plot a kind of Dow Jones curve of the conversation. So this is a real conversation. And in fact, this shows that really it's increasing. This is 0, and so they're becoming more positive. And on average, there's a 5-to-1 positive to negative thing each time they talk. And this is a pretty stable marriage. On the other hand, this is really, when you watch some of these movies, you know, really, you can never talk to the people. But I remember thinking, you know, get rid of them now, or something. Anyway, yeah, of course you can't. Okay, this is a marriage that's on its way down. So what we did was the data. This is the data. We took about 700 of them. We followed them. They did the 15-minute argument, and it could, money, sex, in-laws, you name it, made absolutely no difference. And then what we did is we predicted whether they would get divorced, stay married happily, happily or unhappily. And then we followed them every 2 years. 1 to 2 years, rather. And then we compared what they wrote in the report of their marriage with what we had predicted. The prediction of which couples would get divorced was 94% accurate. In fact, the couples we said would get divorced, we were 100% accurate. What brought the statistics down was those we said, some of them that we said would stay married, but unhappily, some of them got divorced. But even so, the numbers were, I found, really very surprising. And of course, nobody likes to think that their emotions can be so easily categorized. However, for therapy, which is the key thing about this, we thought we'd use this to show couples who came for therapy what the numbers are going to look like when we showed them, the curves rather. So we had 2 actors come, and if they ended up in the first quadrant, they're both happy. And then wives unhappy, and so you go on. And this turned out to be really very, certainly couples, I didn't do any of the therapy, but this is a typical data set that we get from a couple. They start a conversation and it jumps all over the place. And when I first saw this, I thought, yeah, what can we do with that? Well, it turns out it's not really as bad as it was. I mean, I got to the stage where I could look at data like this, and now that couple's, they're in a bit of a mess. But when you analyze it, and so let me just describe what the model is very briefly. It's not very complicated. The husband has an influence on the wife, and T is when she speaks, and so he speaks. The last time she spoke, there's a contribution, and then it depends what she looks like. The mathematical equation is like this for the mathematicians, that W of T is just the number, the score that she gets when she replies after the husband speaks. And this represents the term of how she felt before. And so if she was really mad before, and R is equal to 1, it means it's not easy to change her mind. And so this is a kind of inertia, we call them inertia parameters, and this is what she brings to the marriage. I'll talk about that a bit later. This was the key to really the classification of marriage types. That is the data that we got from the graph I just showed you when you extract the influence function. So we had to evaluate 2 parameters and an influence function for the wife, and the same for the husband. And so what we did is we fitted it to a curve, and then, but that's too complicated for us to work with, so we thought, well, what we'll do is we'll just estimate, make these look like 2 straight lines, and that's what we used. And these are typical data for a couple 1 that I'll mention later. And so what we got was really, this is a typical interaction. Husband says something positive, the wife, his influence is positive. If he says something negative, in this example, his effect is less negative than what he said. And so this ended up being a way to classify. This is a real couple, and what you see is that they want to avoid conflict when something negative is being said. And so what one gets is a theoretical interaction, influence function for different couples. They've got little effect on the negative range, and so what one finds, there were only 5 types of couples, which was really very surprising. And this system we used was what let us classify them. And of these 5 types of marriages, really 3 are unstable, and I'll describe them just briefly in a minute. There's the volatile kind. You know, wild arguments, and then they sort of say something nice. I mean, and that can be stable or unstable. In other words, whether or not they're going to get divorced. But generally it was divorce. There's the validating time. They value this companionate marriage, and the influence function. The influence functions are very clear. They clearly fall into one of these categories. There's then the avoiders. They don't want to. They just avoid, unnecessary confrontation. And the validators, there's the unstable ones, is that they don't want to talk about difficulties, or they're validating. If she says something, that he doesn't agree with, he says, well, you're probably right. So it really boils down to communication. Hostile detached is really the kind of reverse of that. And so, but these are the two main stable marriages. And so this is just the model again. And so if there's no influence, which is what you find in a 15-minute conversation, sometimes they don't affect each other, then you look at that simple model and you work out what the steady state of the wife is when the husband really isn't having much effect. And so there are two parameters, A and R. You do the same for the husband. And this was what really was said. If A is less than 0 and B is less than 0, we predicted divorce. So in other words, when they're not really, in a sense, communicating, they're really both pretty unhappy. And so it was that simple a prediction. And maybe I should add it's not statistical. These are real numbers of real couples. And that's where we got the numbers. And so for therapy, at least one of these things must be positive. R has to be less than 1. Okay, so what we can do is we plot these interaction lines, which says that on that line, the wife is always at the same level in the conversation. You do the same for the husband, and where they intersect, you get the marital steady state. I'm leaving out a lot of the algebra. But basically, if the uninfluenced husband's steady state is positive, and the same with the wife, then you would expect it to have a steady state which is stable and in the positive quadrant where they're both pretty content with it. And of course, when I did some of this and looking back in the library in the Bodleian, I thought that was quite a nice medieval picture of a happy couple. I mean, I don't know what they're kind of fooling around in the bushes here, but that's it. So you can then play the game as what happens if the husband becomes less happy. So he moves down here. And if the husband is then unhappy in the marriage, but the wife is still, and she hasn't, we keep it there, then the steady state starts to become negative. And then if the husband, for example, becomes even more negative, what happens is you end up with no steady state at all, and it's some kind of Calvinistic hell or orgy of mutual satisfaction. I don't know which. But the point is the marriage is totally collapsed. And of course that's a typical Van der Weyden view of Okay, so these are just to show real couples. These are the null clines of a stable marriage where you have a positive steady state. And it doesn't matter where you start the conversation, even if it's low, it ends up at the positive steady state. On the other hand, this type of marriage can end up with 3 steady states, 2 of which are unstable. It doesn't matter where the conversation starts, it always ends up negatively. That couple that I showed the real data for, in fact, that's where the steady state is, and this couple actually got divorced. And their graph had their positives, their steady state both of them were very negative. And that sort of couple is really not, it's not a couple that has got much success for therapy. On the other hand, that one, at least one of them, is positive. And this is a couple that started therapy. It really started it way back in 1998. And this was their discussion before therapy. I mean, it really got very, very negative. And then after therapy, they did the analysis again. They ended up in the first quadrant, stable. And then they did the conversation again. I mean, it might, you know, it might not be joke a minute, but at least one of them is positive. And so they now actually, as I said, their therapy has been immensely successful, and they now have 3 children. And so what we got is really a language to describe human interaction. And it then suggests therapy. They're showing the results of it, and they're showing what they will have to do if they want to try and save that marriage. And as I said, it's all been very, very encouraging. And the predictions, well, I've talked about these already. So it's a new language. It's got really, it's how to deal with human interactions. And I feel that the application of mathematics in the future, biology and medicine, everybody accepts that's fine now. Social sciences and others, I think, are going to be a really growth area. And so it's a rationale for marital interaction. There's only 5 types of marriages. Stable ones are matched interaction styles, and the other ones mismatched styles. And so really, just to finish, I thought my whole philosophy about working in biology, medicine, you know, you call it whatever, it really comes from, you know, I find his approach, his view in one of his essays on this essay here, about he was an immensely practical man. So he said that, as you can read it in truth, and I'm not afraid to confess it, I'd, you know, offer a candle to St. Michael and another to the dragon, if it's going to help. There's a different approach, which I'm afraid is still not uncommon, but I thought rather than quote anybody from the 20th century, I thought I'd go back to Auguste Comte. I mean, I know he's French, I don't mean it that way. And he says, as you can read it, in mathematics we find the primitive source of rationality, and to mathematics must the biologists resort to carry out their researches. That's not a good way to start research collaboration. Then a friend sent me that picture. I was telling him about this in another quote, and so he sent me this picture, and he says, who do you think the stork is? Which wasn't difficult to understand. And then he sent me the next one. And that's maybe a good place to stop. Thank you very much. Thank you. Yes.
I wanted to ask a question, but I have to tell a quick story. The couple had fought bitterly for 40 years, and before getting divorced thought they should have some therapy, and it seemed to work very well. And it was New Year's Eve. They broke out the champagne, and he raised his glass in a toast and said, "I'm wishing you everything you wish me." "What?" she said, "you're starting again?" The question I want to ask you is from the first part of your talk. You showed the diffusion results, and the domain has a big effect in size and boundary. So do you think if by varying things with rather strange boundaries you could have a universal computer?
I'd pass that question to Dr. Davies. Yes.
On your last diagram, that reminds me of the stability diagrams for my power supply designs. But to another subject, if you like the beautiful patterns on that cat, that Joffrey's cat has 36 chromosomes, and it's also too wild to make a pet out of. However, there are a few breeders in this country who breed it with a Bengalized domestic cat of 38 chromosomes, and get a result called a safari cat with 37 chromosomes and some very interesting patterns on it. And if you can afford the $7,000 fee to the breeder, you could have that nice cat, which will come when you call it. You can train it to go to the toilet, and it does everything a dog and a cat can do, and much more, with 37 chromosomes. So you might, it's a rather obscure fact, but I thought you might be interested in that. So thank you very much.
Oh, okay. Thank you. That's it. That's okay.
So I hate to say something which may sound obvious, but going back to your gliomas.
Going back to, sorry? I didn't hear what you said.
With respect to gliomas, now obviously there's a great disadvantage to cutting away any more of the brain than is necessary, but it seems that your models show that surgery would be more successful if they were to cut away more than they can see. Based on your models of how much the tumor has actually expanded beyond the visible range. Has that had any clinical effect? Has there been any sort of reaction about that?
Well, no, that was what I meant about these different curves. Depending on the parameter values, if the surgeons actually took note of that, then they might cut out a little bit more and would give longer life expectancy. But surgeons are not like neuropathologists. They tend not to be interested. I mean, that's a very personal view, but they really they see things very differently. And now a lot of this, there's a group in the hospital in Seattle where what they're now doing are quantifying things like that. Not only that, the model lets you quantify really chemotherapy, radiation, and because we know what the parameter values are of the individual patient, it means that we can quantify the efficacy before it's tried. And some of the patients decide it's just not worth it. And so in this sort of thing, it's really providing information that is definitely medically relevant, and there's quite a study going on now on this.
Thank you very much. Could you describe to me, please, and indeed if it's going to bore everybody else here, let's talk in a corner, but I would be very interested to know how you set up the cameras and you are satisfied that what you are getting is an absolutely honest interaction.
I think there's no doubt that the visual expressions are used when the people quantify what the people are saying. Setting up the cameras we didn't even think of it at all very much. It was the listening and the scoring that was all we used in the model. And when you say, how do we know in a 15-minute conversation of a problem of contention, within almost at most 2 minutes, they're at it. And really, they just completely forget the fact that they're being observed, they're being filmed, they're being, just everything about them. There even be, in fact, to begin with, we used to take their blood pressure. And one couple, he scared me silly. During the argument, his blood pressure kept going down. And when I asked where they met they had met in some paddy wagon going to prison. But this guy was really very scary. And so one can get too influenced by looking at the movie rather than just taking the data. And so I ended up really just looking at the data.